Welcome to The Explainer. Today we're diving headfirst into one of the wildest, grandest evolutionary mysteries out there. We are going to trace the very architecture of human life all the way back to an invisible, ancient world. We've always wondered, how did the microtubules that literally give our cells their structure actually evolve? Well, the secret lies hidden in a group of fascinating deep-sea microbes. We're going to explore exactly how these ancient organisms hold the missing blueprint to the very structures keeping our own human cells intact. So get ready, because the clues we're about to uncover bridge a gap of billions of years, and they reveal the absolute sheer power of primitive life. Think about it. Every single cell in your body right now relies on an incredibly complex internal scaffolding just to survive. Seriously, imagine for a second what a cell would be without a skeleton. It would just be this formless, deflated blob. It wouldn't be able to move or divide or really do anything at all. But that brings us to the ultimate missing link mystery. How in the world did evolution build such a microscopic masterpiece? Why was this cytoskeleton born in the first place? And what did its earliest blueprint look like long before complex life as we know it even existed? Here is our roadmap for today. We'll kick things off with the cytoskeleton mystery. Then we'll meet our primary suspect, the Asgard archaea. From there, we'll dig into the microscopic evidence of minimal microtubules, watch this machinery physically deform membranes, and finally, we'll tie it all together with the roots of life and what it means for future tech. All right, Part 1, the mystery of cytoskeleton evolution. Before we travel back in time, let's make sure we have our biological foundation set. Inside your cells, you've got these incredible, dynamic structures called microtubules. The best way to picture them is like a cross between a rigid skeleton that gives a cell its shape and a super busy transit system of internal rails. They're built entirely out of these proteins called alpha and beta tubulin. And without them, the whole bustling inner life of a cell would just grind to a halt. Nutrients couldn't be delivered, critical parts of the cell couldn't be positioned properly, signaling molecules would get totally lost, and your cells could never successfully divide. They are literally the fundamental highways of biological life. Now, modern eukaryotic microtubules, like the ones found in humans and other complex organisms, are seriously intricate. They are typically built from 13 individual fiber lines called protofilaments, and these are arranged in a thick, continuous ring. Because of this incredibly robust 13-filament design, they're super rigid. They can withstand strong pushing forces, usually around 3 to 4 piconewtons, and they also have this vital feature called dynamic instability. That basically means they aren't just static poles, they're constantly, rapidly growing and shrinking to explore the space inside the cell, all driven by chemical energy. But here's the big question. How did a structure as thick and complex as this 13-filament ring just suddenly appear in the evolutionary timeline? Moving to Part 2, the true identity of Asgard archaea. To answer that evolutionary question, we have to introduce our prime suspect. 2.7 billion. That is a mind-boggling number of years, right? But that's roughly when a specific group of microbes known as Asgard archaea, specifically the Heimdall archaeota group, are thought to have emerged. To put that into perspective, the Earth itself was vastly different back then, and complex life was still an eternity away. Yet these ancient organisms are widely considered to be the absolute closest known relatives to complex cellular life. They act as this profound evolutionary bridge, connecting simple, single-celled bacteria to the massive, complex organisms like us that eventually inherited the Earth. But here's the catch. Studying these profound ancestors is notoriously difficult. These archaea live in extreme deep-sea environments. The immense pressure and unique conditions make actually growing them in a lab practically impossible. So, an international team of scientists pulled off some truly brilliant evolutionary detective work. First, they extracted metagenomic DNA straight out of the mud in the San Francisco Bay estuary. Then they found two tubulin-like genes sitting right next to each other, and ingeniously inserted them into E. coli bacteria. By using the E. coli as a sort of microscopic factory to artificially pump out these ancient proteins, the scientists bypassed the whole deep-sea pressure problem. They could finally purify them and study their behavior right there in a simple test tube. Which brings us to Part 3, discovery of primitive minimal microtubules. Because what happened next in those test tubes completely blew the lid off the origin of the cytoskeleton. The contrast here is just incredible. Remember how modern microtubules need that massive, rigid 13 protofilament ring to work in our large cells? Well, the ancient proteins from the Asgard archaea told a totally different story. They self-assembled into primitive high microtubules made of just 4 protofilaments. 4. It's a remarkably thin, highly flexible, and beautifully minimalist structure. It is, quite literally, the stripped-down, bare-bones ancestor of our own cellular skeleton. It gives us a perfect, crystal-clear snapshot of what early structural biology actually looked like. But here is the absolute craziest part. Despite being this minimalist 4-filament design, these ancient archaeal tubes share 4 crucial functional properties with our complex cells. First, they form paired alpha and beta dimers, exactly like ours do. Second, they form distinct hollow tubular structures. Third, they show that same vital dynamic instability, growing and shrinking all on their own using chemical energy. And fourth, amazingly, they generate highly effective physical pushing force. The core mechanics of the human skeleton were already entirely functional in this simple, flexible microbe billions of years ago. It's the exact same fundamental technology, just scaled way down. Part 4. Incredible power deforming the membrane. Scientists didn't just want a static look at this. They wanted to see the power of Asgard archaea actually in action. Using advanced cryo-electron microscopy, researchers mapped out the incredibly precise molecular structure of these ancient 4-protofilament Heim microtubules. But the real magic happens when they're put to work. The researchers took these purified ancient proteins and placed them inside artificial cell membranes, known as liposomes, to simulate a living environment. And guess what happened? As the minimal microtubules assembled and bundled together inside, they generated enough physical force to literally push and stretch the artificial cell membrane from the inside out. They created these distinct glowing spikes, visually proving their raw mechanical power. The efficiency of this ancient machinery is just staggering. When they were placed inside artificial liposomes measuring 10 to 20 micrometers, over 70% of the artificial cells deformed into distinct ellipses, entirely due to the pushing power of the ancient microtubules. Now, sure, a single primitive tube only generates about 0.08 piconewtons of force, which is way less than a modern cell's 3 to 4 piconewtons. But they make up for it with teamwork. When these flexible tubes bundled together, they produced a collective force of 0.5 piconewtons. This completely proves the immense and surprising power of Asgard archaea. It shows that structural bundling was the evolutionary key to shaping and manipulating cellular boundaries. Finally, Part 5, Roots of Life and Future Technology. We've seen the evidence, we've seen the raw power, but what does this actually tell us about the ultimate roots of complex life? The logic of evolutionary progression becomes perfectly clear when we step back and look at why the cytoskeleton was born in this specific way. Billions of years ago, small primitive archaeal cells only needed minimal, highly flexible 4-filament structures. It was metabolically cheap to produce and perfectly effective for a tiny, tiny space. But as evolution marched on and eukaryotic cells grew vastly larger and more complex, they needed something that could bridge larger internal distances without just buckling under the pressure. So the metabolic budget expanded, and the thicker, rigid 13-filament tube was evolutionarily selected. It's a beautiful, logical answer to the mystery of microtubule evolution. One of the researchers noted that recreating this mechanism of membrane deformation with just a few proteins directly leads to applications in artificial cells and synthetic biology. This isn't just some dusty history lesson about ancient biology, guys. It is a profound blueprint for tomorrow. By understanding how to control the thickness, the stiffness, and the collective pushing power of these minimalist tubes, scientists now possess a truly foundational technology. We are literally learning how to design customized molecular machines and tightly control the shape and movement of artificial cells entirely from the molecular level up. We started out looking deep into the mud of the San Francisco Bay, and now we're looking at the absolute frontier of synthetic biology. By mastering the 2.7 billion-year-old minimalist blueprint of the Asgard archaea, we have to ask ourselves, what kinds of custom, self-shaping artificial cells are we going to build next? It is wild to think that the tiny, ancient ancestors of our own cellular skeleton might just become the master architects of our future technologies. Thanks for joining me on this explainer, and I'll see you in the next one.